Hello everyone, I am Dr. Srinivas. In this module, you will learn about molecular techniques part 1. Under molecular techniques, you need to know about recombinant DNA technologies, DNA cloning, PCR techniques, blotting techniques, gene therapy, and few points about DNA polymorphism, SNP, VNTR and RFLP, and genomic and cDNA libraries, DNA microarray, and human genome project. What is recombinant DNA technology? It is going directly into the DNA molecule for information and manipulating the DNA sequence by constructing a chimeric molecule through genetic engineering. Here you can see this is the chromosomes, and chromosome is highly coiled into chromatids and it is organized into DNA organizational structure. Here you can see this is the chromosome which is in coiled form nucleosomes, and they are wound around protein called histones, and ultimately we have DNA double helix. So, in order to get the information out of this chromosome, we have to go into the DNA double helix and get those information, and that information flow is from the DNA into the RNA and protein, and this is called central dogma. So, central dogma, there is information flow from DNA into mRNA and then into protein, and it is rigidly controlled by number of processes. And in molecular technique, we will understand all those processes where this central dogma can be exploited for our benefit. So, in general, the information coming from DNA is stored as genomics. Information coming from mRNA is described as transcriptomics. Information which is studied in proteins are denoted as proteomics. Using those informations, we will be able to do what is called as RDT, that is recombinant DNA technology. So, what and how this RDT is done? This is a technique of joining different sequences of DNA, forming a new combination which is called recombinant DNA, and this technique recognizes normal and pathogenic genetic variations. For that, DNA must be isolated from appropriate source in appropriate amount, and it is made available for the study with the help of a technique called PCR, polymerase chain reaction. So, the technique of isolating and amplifying DNA for study as well as manipulating involves use of 1. The restriction enzyme, 2. the cloning vectors, 3rd. the PCR technique, 4th. the gel electrophoresis, 5th. the blotting technique, and then 6th. the probes for hybridization into appropriate target DNA sequences. With the help of RDT, we will be able to analyze all the expressed genes in a cell using a assay called gene chip, which can lead to the profiling of a normal versus diseased cell. To understand RDT, we need to understand an important enzyme, that is restriction enzyme. Restriction enzymes are enzymes seen in bacteria. They are produced inside the bacteria to fight off viral DNA which are injected into the bacteria into their viral genome with the help of bacteriophage. So, bacteriophage are the viruses which infects bacteria. So, the bacteria, in order to protect its own cell, produces an enzyme called restriction enzyme, restriction enzyme act on those viral DNA materials or RNA material and cuts them, so that this viral DNA is not integrated into the bacterial DNA and prevents the host from getting destroyed. So, this enzymes we make use to make new combinations of DNA sequence, which is nothing but recombinant DNA. So, what are the application of recombinant DNA technology? This technique is used to prevent and as well as treat diseases. For example, a recombinant DNA technique is developed to produce human insulin for treatment of diabetes, to produce factor 8 for the treatment of hemophilia, to produce vaccines as well as prevention of hepatitis, and also for diagnostic purpose like ELISA for production of antibody as well as monoclonal antibody treatment as a part of cancer therapy called immunotherapy. It can be used to develop gene therapy, which is in experimental phase, and it's possible to develop genes or replace a defective genes, and it is having ability to analyze the whole genome and proteomes and help in establishing an expressed protein, a defective protein, or a protein which is inhibiting other normal wild type of proteins. So, for all those applications we require recombinant DNA technology. So, best known example is humanin, that is human insulin. Initially for the diabetic patient the pig insulin was used. So, we know that pig insulin is well tolerated in humans, but still there is antigenic exposure might lead to development of antibodies against the pig insulin, which is called procaine insulin. So, RDT has made it possible to generate an insulin from human genes. So, how it was developed? Briefly it was described here. So, we take a synthetic gene fragment which code for insulin, insert it into a plasmid. Now, this plasmid become a recombinant plasmid, and the plasmid is injected into the bacterial cell, and the bacterial cell is made to divide, and they produce variety of daughter cells having human insulin gene in their plasmid. Once the plasmid is expressed, insulin is also expressed. Those insulin is purified and packed as human insulin. This is possible only because of recombinant DNA technology. So, in recombinant DNA technology, usually we come across a term called chimera. Chimera is basically a combination of two different organisms, like you can see in this picture. There is human with a lion face, there is a tiger with a horse face. So, this kind of combinations, if we term it as chimera. In RDT also there will be a bacterial gene, inside that there will be a human gene. So, a human with bacterial gene is a chimeric DNA sequence. So, in DNA recombinant technology we join different DNA sequence to produce a new combination, which is called recombinant DNA technology. For that we use an enzyme, restriction enzyme, restriction enzyme has a property to identify a specific segment of DNA and cut in a palindromic sequence on both the strand. Here you can see an example of restriction enzyme, that is ECO R1. ECO R1 is coming from E. coli and it is cutting, it is recognizing this sequence, that is GAATTC. If you read this, it will be same from the other side, GAATTC. This is called palindromic sequence. It recognizes this particular sequence, cut the DNA strand and produce something called as sticky ends. So once it cuts, it will deliver a space here. We can insert our gene of interest in this sticky area and with the help of DNA ligase, ligate it with the bacterial DNA and produce a recombinant DNA segment. So this is what restriction enzymes does. It recognizes a particular sequence, cut the strand and with the help of DNA ligase, we can reseal the strand producing a recombinant DNA. So there are different types of restriction enzyme. Here you can see a restriction enzyme called HIND3 digests a particular DNA segment producing a 5' protruding sticky end, whereas PST1 will produce 3' hanging sticky end, whereas ECO R5 produces a blunt end. So a restriction enzyme can produce sticky end or a blunt end, and there are variety of application where we use blunt end, or in some cases we want to use a 3' protruding end or a 5' protruding end. And the best example of how RDT can be used as a diagnostic tool is in diagnosis of sickle cell anemia. Here you can see a normal sickle cell anemia DNA after mutation is converted to from A to T. There is conversion from adenine to thymidine. So GAG has become GTG. This change in the nucleotide leads to change in the RNA, and the change in RNA produces change in the amino acid from glutamate to valine, and the change in a amino acid will convert the normal protein into a mutant protein, and this mutant protein causes sticky patch and ultimately lead to production of a sickle shaped RBCs, and these sickle shaped RBCs will accumulate in the blood vessels causing Rolex formation and blocks the blood supply, leading to sickle cell crisis. So this particular disease can be diagnosed using recombinant DNA technology, specifically restriction fragment length polymorphism. So sickle cell anemia is an example of point mutation where a glutamate residue is converted into valine residue because of change in the DNA from GAG to GTG. So this change will lead to abolition of a restriction site in the beta chain. In this you can see there is a beta globulin chain gene. So there is 3 restriction site for restriction enzyme MST2. So MST2 in a normal beta globulin chain gene has 3 restriction site in the beta chain. So the MST2 will cut the DNA segment into two fragments. So here you can see in this gel electrophoretic picture, there is normal adult hemoglobin. So after treatment with restriction enzyme, there is two fragments which are produced. If there is a restriction point mutation, it abolishes a restriction site, one of the restriction site. It is seen in this diagram GAG. If there is a T, that restriction site is abolished. That means the MST2 now can't recognize this site. Once the gene is subjected for the digestion, it won't be able to cut into two fragments but into only one fragment. Here you can see when the person is having sickle cell disease, there will be only one fragment which is visible in the gel electrophoresis. If the person is a carrier of sickle cell disease, that means one coming from the parent is normal, maternal parent, father side is normal, one coming from the mother side might be a carrier of a sickle cell. So, the restriction enzyme now will cut the normal paternal side chromosome gene into two fragment, but maternal side into only one fragment. Gel electrophoresis now will reflect three bands. So, this type of pattern, if we call it as sickle cell triad. So, if a three bands are seen, it is sickle cell triad. If only one big band is seen, it is called sickle cell disease. If shorter two bands are seen, that is a normal type of hemoglobin picture. So, using recombinant DNA technology and an enzyme MST2, we are able to diagnose a single point mutation in single beta chain gene, and also we are able to identify whether it is disease or a carrier state. So, RDT using MST restriction enzyme can recognize a sequence having CCTNAGG. If there is any change in this sequence, the enzyme is not able to recognize, hence we will be able to having fragment length differences. So, that is the reason why this technique is called restriction fragment length polymorphism or RFLP. Using the same technique, we will be able to diagnose cystic fibrosis as well as phenylketonuria. In cystic fibrosis gene, it is the protein CFTR which is mutated, and the mutation can be diagnosed easily using RFLP technique. Coming to second important technique under molecular biology, it is called cloning. Cloning is a technique used to make an exact genetic copy of a living things. So, genes, cells, tissues and even whole animal can be cloned. Some clones are already exist in the nature, like single cell organism like bacteria make exactly the same copy of themselves each time when they reproduce, but higher organism it is very difficult to produce exact same copy. But a well known example of cloning is Dolly. Dolly is the first sheep to be cloned, and this is the cloning procedure. how you can produce your own copy. In this procedure, what they do is they will take the egg of the donor sheep, they will take out all the nucleic acid materials from the egg, which is called enucleation, and from somatic cell donor. So, if you want to clone this sheep, you take the donor cell, take out the nucleic acid or genetic material, inject those genetic material into the cytoplasm of the recipient donor egg, and you produce a embryo which is called reconstructed embryo. Grow those embryo in the petri dish called blastocyst, transfer that into a foster mother, and the foster mother will give rise to exact same copy of the cells from the donor, and the dolly was the first sheep to be cloned by this technique. But still there are so many abnormalities which were seen in the dollies, like the dolly had autoimmune diseases, arthritis. So, there are lot more things to be studied during cloning. It may not be a simple transfer of genetic material and growing a similar clone which you want. So, to construct a recombinant DNA technology, DNA molecule, what you have to do is you require a genetic material as a starting point. You take the genetic material, use a restriction enzyme, cut, cut those into smaller pieces, isolate your gene of interest, and these all can happen within a test tube. Once you isolate your gene of interest, take out a vector, in this case a plasmid vector, use the same restriction enzyme to cut it, cut open the plasmid vector, insert your gene of interest into the cleavage site, using DNA ligase seal off those plasmid site, and you have a plasmid chimera which is recombinant DNA. Once the recombinant DNA is inserted, it is called chimera. Now you need to insert this recombinant DNA into a bacteria for the multiplication as well as expression. So, you are inserting this recombinant DNA into the bacteria. This insertion can be done using heat shock, nanoparticles, cold shock. So, the plasmid now with our gene of interest is entered into the bacteria, and the bacteria start multiplying along with our gene of interest. So, you can collect those materials which is having gene of interest. How to collect the material or the cells, how to identify those cells which is carrying our gene of interest. It is done by using a marker called antibiotic resistant. So, these plasmid vectors will carry an antibiotic resistant region. So, any bacteria which take up the plasmid, recombinant plasmid, will also acquire a resistance for the antibiotics used. So, once in a petri dish, if you introduce an antibiotics, all other bacteria without this gene will die off, and only those colonies having the resistant gene will be able to multiply. And this is an indication like the plasmid has been taken up by those bacteria and they are living. So, there are variety of vectors by which our gene of DNA can be transferred into the bacteria. They are called fudge lambda-phage, cosmids, plasmid artificial chromosome, bacterial artificial chromosome, yeast artificial chromosome. These vectors are limited by their package size or insert size, whereas the lambda-phage can carry only a small fragment like 25 to 5 to 25 kilobase of DNA materials, whereas yeast artificial chromosome can carry up to 2000 kilobase pair of nucleotide or DNA material. So, this is about restriction enzyme and how it is can be used for developing a recombinant DNA. But what is the use of recombinant DNA? Then there comes an expression vector, and here you can see there are mapping for variety of restriction site. So, using any of these restriction enzyme, we can open up this expression vector, introduce our gene of interest and take this recombinant DNA, insert into the bacteria and make the bacteria produce our protein, in this case insulin, and produce human insulin. This is one application of recombinant DNA technology. The next important application of RDT is gene therapy, and the best well known example of gene therapy is ADA deficiency. ADA deficiency is adenosine deaminase deficiency. This is an important enzyme in purine nucleotide metabolism, especially important for immune system. So, deficiency of this enzyme lead to a disease called SCID. SCID stands for severe combined immunodeficiency. So, there is both B cell and T cell deficiency in this patient, ultimately leading to total immune failure, and they are prone for all infection and hence to be kept in a bubble. So, they are called bubble babies, and in these cases we need to supply this defective enzyme called ADA through gene therapy. This is the bubble baby. They are always kept inside a bubble so that they are not able to interact with the outside environment, because they are not tolerant for any of the bacteria, viruses, fungus, because both B cell and T cell immune system are lost, hence they are called bubble baby syndrome. And adenosine deaminase is an enzyme responsible for purine nucleotide, and deficiency of this enzyme will lead to deficiency in the nucleic acid synthesis, leading to both B cell and T cell damage, and they are prone for fungal, bacterial, viral as well as protozoal infection, especially pneumocystic pneumonia, and they are always failure to thrive. So, this patient can be treated by supplying adenosine deaminase into the hemopoietic stem cell, and this case is usually autosomal recessive and it is present in chromosome 20. How we can deliver such an enzyme into a particular type of cell, specific type of cell? This is accomplished using gene therapy. So, a gene therapy of ADA deficiency is done like this. You collect those cells from the patient, in this case T cells, you grow the T cells, then using restriction enzyme you prepare a recombinant DNA where a normal human ADA gene is present, you infect the grown T cell with the new DNA, that is recombinant DNA, and the infected cell will now carry a normal human ADA and it gets corrected. You amplify those cells and then infuse back into the same patient. Now the patient body start producing ADA and all their immune dysfunction is been able to treat. So this is the first disease where gene therapy was successfully achieved. So the baby which you saw earlier now is at 5 years and still living in the cell waiting for ADA gene therapy to happen. So a gene therapy involves isolating a normal gene, inserting them into the disease cell so that a normal gene is expressed and permitting the disease cell to return to a normal state. So this is called ex vivo gene therapy, where we isolated a defective gene, grown those cells, infected with a recombinant DNA or RNA, and this causes a corrected cell to be formed and recovered cell are transfused back into the cell. Now they are producing a normal gene expression. So this is in short the utility of gene therapy, and gene therapy can also be used as a disease drug screening, toxicity testing, or to produce a disease modeling. So the next aspect of molecular technique is gene editing. Gene editing is different from gene therapy. Here a normal gene inside a cell is edited or removed from the DNA. So this is necessary because for diseases such as Duchenne muscular dystrophy, so that is due to a defective production of dystrophin gene. We can't isolate all the muscles, transfuse them with the corrected genes so that they start producing dystrophin. So in some cases we need to edit the gene which is present inside the cell itself. So for that particular purpose a technique called CRISPR-Cas9 is used. CRISPR-Cas9 uses a guide RNA which will recognize our target gene, and with the help of an enzyme called Cas9 it cuts the targeted gene and a replacement gene is replaced, and with the help of DNA ligase it is ligated back. This is called gene editing. So what are the applications of recombinant DNA technology? Here we can use it for developing hybrid food stock. BT cottons, BT brinjal are such example which are disease resistant disease resistant plants and high yielding, and we can use it for gene therapy, use it in human germline so that we can modify the embryo and produce superhumans, or you can have a designer organisms which are not ethically not allowed in most of the countries. So the next aspect you have to learn in molecular technique is probes. So these probes are small, around 25 to 2000 base pair length only, a fragment of DNA or RNA. They are complementary to the sequence of gene which is our interest, and they can be labeled using a chemical or fluorescent and used for detection purposes. Initially we used to use radioactive probes. Now radioactive probes are not used, they are replaced mostly by chemiluminescence probe. The radioactive probe initially used were phosphorus 32 and DIG, and they are attached with the probes, and the probes have a sequence similarity or complementarity to the gene of interest. Once they combined in a form hybridize, this probe would give out a signal, and detection of those signals will be read out and a particular disease pattern is recognized. And these probes work on a principle of hybridization, and we have variety of probes like in southern blotting the probe is against DNA and the probe is made up of nucleic acid. Northern blot is used to detect RNA and the probe is again nucleic acid. In western blot the probe is protein and the detection is again protein. So, in southwestern, which is basically used for detection of transcription factor, where the probe is DNA and the target are protein. So, northern blot is a technique for detection of RNA. How this technique is being done? We extract RNA from the sample and run it under electrophoresis. RNA are separated based on the size. Now they are transferred into a membrane, usually a nitrocellulose membrane, and a probe which are labeled are poured onto the nitrocellulose membrane, and wherever our target RNA is present, the probes will go and bind to them and they will give out a signal readout, which are read out. Initially, if it was radioactive, it was read out in X-ray film. Now it is chemiluminescence, we read out in a gel doc or gel documentation system. Similarly, for western blotting, there is a probe which is nothing but an antibody produced against the protein of interest. This antibody is tagged with a chemical or an enzyme which can convert a chemical into a chemiluminescence, and the light which is emerging is detected in gel doc system. So, the technique is similar to your ELISA technique, where antigen is bound, antigen antibody reaction is exploited. The antigen here is the protein of interest, antibody is the probe which is labeled with a chemiluminescence or a substrate, chemical substrate. Here the major difference is the electrophoresis is run on SDS page, which is nothing but polyacrylamide gel electrophoresis, where all protein charges are neutralized and in uniform negative charge is applied. So, the protein are separate based on only molecular weight. Whereas in southern blotting, it is used to detect DNA. The technique is similar. The whole genomic DNA is extracted and using restriction endonuclease enzyme, the genomic DNA is cut into smaller size fragment and they are run under gel electrophoresis and they are separated based on their molecular weight. After separation, they are blotted or transferred into a nitrocellulose membrane. Once it is transferred using a probe, we will be able to detect a particular gene segment is present or amplified or defective, whatever may be the study objective. So, the technique of detecting DNA is called southern blotting, technique of detecting RNA is called northern blotting, and technique of detecting protein is called western blotting. And the procedure is same. You run the sample on electrophoresis, transfer into a nitrocellulose membrane and apply a probes to detect or visualize the target molecule. Southwestern is specific for transcription factor, where the probe is protein, target is protein and probe is DNA. So, the protein which are binding the DNA fragments, that is transcription factor, are the target molecules for the study using Southwestern. So, the procedure: separate the protein using SDS page, renature the protein by removing SDS in the presence of urea, transfer them onto the membrane. Then now the genomic DNA of interest is digested using restriction enzyme which are labeled. Then add a separated protein to it to see which transcription factor binds to that particular genetic segment. The next important technique under molecular biology is FISH. FISH is nothing but fluorescent in situ hybridization. Here the DNA probe is labeled with fluorescent dye, and this fluorescent dye will bind to those segment of DNA which has this probe similarity, complementarity. Once it binds to those region, it gives a fluorescent light and it is detected under fluorescent camera. This will ensure which region of chromosome is been attached with the probe under study. So, a patient DNA is taken, it is denatured, a probe with fluorescent label is used, and with the hybridization technique it goes and binds to it, and it is used to diagnose variety of diseases like Down syndrome. We know that in Down syndrome there will be 3 pairs of chromosome 21. So, for a particular gene segment in the chromosome 21 is probed, and if we get 3 signals like we are getting in this picture, that suggests the patient is having Down syndrome, and it can be used for Edward, Patau. So, a Down syndrome, here you can see in a karyotypic of Down syndrome you have 3 pairs of chromosome 21. Similarly, in Edward syndrome there will be 3 pair of chromosome 18. They all can be probed using FISH, fluorescent in situ hybridization. Similarly, in Turner syndrome there will be absence of one X chromosome, that also can be detected using FISH, where we can see only one X in a female patient, leading to a diagnosis of Turner syndrome. There is something called MFISH, where we can probe for multiple diseases in a single go by using a multicolor fluorescence probe. So, if you want to diagnose all disorders of chromosome, known chromosome like 21 chromosome anomaly, 18 chromosome anomaly, 13 or Klinefelters or Turners. So, use a probes with different fluorescent and it will give a picture like this, and this technique is called MFISH. So, molecular biology technique is versatile and it is evolving day by day and is used for variety of diseases, both for diagnosis as well as for the treatment purpose. So, hope you have learnt molecular biology techniques and will continue with the further techniques in part 2. Thank you.